Okay, so this is the morphology lecture, which I guess maybe I'll splice these videos up. So these are just notes about lab, important basically. So we'll start with the zebrafish life cycle, right? So this kind of cartoon, if you want to call it that, has been pulled up throughout a few slideshows, I think, throughout the semester, right? And it's basically just the life cycle of a zebrafish, right? So 30 minutes after fertilization, we have all the developmental stages following zygote, zygote, and cellular cleavage. So, I mean, the figure is literally 30 minutes after fertilization, right? So all this division happens, cell specification happens, gastrulation and epiboly. And then I can't see behind the phone. And then down here, we have organogenesis. Oh, I should have pointed out the time frame of this. All right, so 0 hours to 16 hours. And then over here, shortly after organogenesis occurs, day 1, after fertilization, day 2. So this is if our zebrafish larvae were free swimming and not in their, essentially the chorions, they would look kind of like this, right? They still rely on their egg yolks from day 2 to, I think, day 5, 6, or 7. But I'm pointing out day 2 because that's when we basically start exposing our zebrafish to our environmental toxin, right? Ethanol in this case. And then, you know, they'll continue growing and maturing. And then this, again, is a cartoon of what they look like at day 90, which is far beyond anything we see in class. And no real questions about that. So that was a cartoon. These are, you know, slightly nicer because they're microscopic photos. They're like light contrast microscopic images used. That pretty much runs the whole developmental process, right? So zygote, fertilized, 2 cells, 4 cells, 8 cells, 16 cells, the whole gambit all the way up until, you know, 24 hours, 28 hours, 33 hours, 36 hours, and then 40 hours again, which is when we start exposing our zebrafish to our environmental toxin. Everyone good? Okay. And so this is a little bit of zebrafish anatomy, essentially. You know, if you guys haven't come across it yet in all the primary research articles you're reading. So this is our, go back. This is our, like, I mean, this looks like 24 hours basically. So 24 hours, you can see where the eye will develop in this. It says right there, sorry, 29 hours. So you can see where the eye is initially going to start to form, where the forebrain will be, where the midbrain is starting to develop by the look of it. You know, these are just light contrast images. So it's not like they have resolution, or it's not like you can see labeled neurons or anything like that. Hindbrain, neural tube, notochords, cell mites, so cell, you know, non-neural cell bodies, and then floor plate. Down here at 48 hours, again, when we start exposing our zebrafish to ethanol, we can see that the heart has started to form, hatching gland, or basically their yolk where they're going to keep getting nutrients from, the developing pectoral fin, neurocell, floor plate, horizontal myoseptum, and fin, right? Someone's at my driveway. And then 5 days, which is a day or two before we start to see them in class, right? So at this point, we can see the eye structure more clearly. If you look closely, you can see the jaw, gills, liver, ear, the swim bladder, which some of you guys chose to quantify information about, yolk sac stripe, horizontal myoseptum, intestines, ventral stripe, and dorsal stripe, right? So some basic zebrafish anatomy if you guys haven't come across it before. And so how do I want to relate this? I don't know. This is hard to do not in a lecture, but so basically the topic of this week's investigation or data collection is to look at morphological differences induced from our environmental toxin. And some of you guys have collected behavior data, right? And so I use these examples from primary research articles looking at opioid withdrawal in mice, just to emphasize how powerful and useful behavioral information can be to collect, right? So when I was an undergrad, I worked in a lab that tested different aspects of opioid pharmacology. So one of the things we tested was withdrawal from physical dependence on opioids. So in the experiment from these graphs, essentially, we deliver relatively high doses of opioids to mice. They become physically dependent on it. And then after a certain number of days, after a certain number of days, we basically precipitate withdrawal from those opioids, right? So we have physically dependent mice, and we're going to send them into opioid withdrawal. So over here, we have saline-treated mice, morphine-treated mice, and then this experimental compound that I brought up a few times throughout class, MMP 2200. Right, so this withdrawal is precipitated by giving the mice naloxone, or Narcan, as some of you guys might know it. So mice that have been treated with saline for, again, however long this is, whether it's 24, 48 hours or 5 days, the mice that receive saline and then are injected with naloxone don't respond. So they don't jump at all. Like you inject these animals with naloxone and they do nothing. Animals that have received morphine for a long period of time, after delivering naloxone, they actually jump. They like jump up like popcorn. And the number of jumps within a fixed amount of time, whether it's 1 minute, 2 minutes, 5 minutes, corresponds to how severe, or we use it as a measurement of how severe the withdrawal episode they're going through. So like with morphine, we get a lot of jumps, right? Especially compared to saline. So these mice are physically dependent on morphine. We send them through withdrawal and they jump a lot. Whereas our experimental compound over here, MMP 2200, doesn't induce as many jumps, right? So it induces a few, but not as many. So one way you can interpret this data is that the withdrawal experience that the mice are going through isn't as severe. Right? And again, this is really just meant to show you that there are valuable things you can glean when you choose the right behavior to observe. And this, I guess you could correlate this to morphology. So when we send our mice through withdrawal, they typically urinate and defecate quite a bit. So we also measure mean body weight loss, right? So the animals that were treated with saline might lose roughly 0.3 grams. The animals treated with morphine, so again, they're physically dependent on opioids. When we send them through withdrawal, they lose 1.2 grams of body weight. And when these mice only weigh 20 grams, they're losing close to 5% of their body weight over the course of 2 minutes through defecation and urination, right? Basically a severe stress response. And then again, our experimental compound over here, they lose, I would imagine these aren't significantly different. So they also lose some body weight. And this is locomotor assay data. If you guys want to know more about this, just feel free to ask me. But basically, we have a tool that allows us to monitor how mice move. And they move differently when they're exposed to opioids. If you have questions about this, feel free to ask me. This really emphasizes the point of why you collect behavioral data and the power you can collect with behavioral data. Right? And then so this is more morphology linked. So one of the pieces of work I was involved in as a tech lab manager grad student was cancer-induced bone remodeling in rodents, right? So over here we have rat x-rays, basically x-rays of rat femur, tibia, and everything else in the ilium sacrum region here. But essentially, in this study, we were looking at how severe bone remodeling occurs after these animals have basically had, these guys had breast cancer cells essentially metastasize to their bones and start remodeling their bones, right? So this is meant to show the power of collecting morphological data and emphasize some of the caveats of morphological data. So I mentioned this in the protocol, and I mentioned it to all you guys, right? So what we want to quantify here is how severe how severe the bone degradation these animals have occurred or have experienced, right? We want to quantify that. But if you quickly look at all these things, maybe you're looking and you're saying that's pretty subjective. It's pretty subjective, right? And that's fine, because making these observations can be fairly subjective, which is why you don't just look at a bone and say, yep, that bone's good, and yep, that bone's bad. One of the things you can do is make a qualitative rating scale where you define a scale, right? So on this system, we had a 4-point scale, 0, 1, 2, 3, and we essentially set up this system, right? So bones that would get a rating of 0 typically look like tumor-free controls, right? So we have good, consistent bone density here, right? We don't have cortical bone erosion, and we don't see like any pitting in the non-cortical bone. Whereas over here, right, so this is the unaffected limb of an animal, and this is the affected limb of an animal shortly into disease progression. So over here, if you just look quickly, you can see that there's some maldeposition going on here potentially. You can see some darker spots. So we would call this like some essentially moth-eaten bones. So we are starting to see some kind of bone remodeling, right? And again, the difference between this and this is fairly subjective. But if you look at enough bones and you adhere to the scaling system, you get used to like, I don't know what that notification is about. You get used to looking at them, and a healthy bone looks different than even a slightly remodeled bone. So this bone is an example of what a rating 1 would look like. And then over here, this bone would receive a rating of 2, right? So we are remodeling a bone. So we have basically cortical degradation of bone over here, and these severe pits within the center of the tibia. So we would call this remodeling with pits. And then our last rating 3, right, the most severe portion of, or the most severe type of observations we would make is a bone that has undergone remodeling with pits and fracture. So if you look, again, at this point, this is a fracture completely through the cortical bone on this limb, right? We have severe pitting. And, I've seen bones worse than this, because you don't know how bad the bone is until you take an x-ray and observe it. But, this is severe bone remodeling and fracture. So this is a severely damaged bone from cancer. And so the point here is, again, you can develop a rating scale like this to quantitate otherwise qualitative information. You just have to define your criteria, and then, typically you would present representative images just like we have here, to show this is what a 0 bone looks like. This is what a 1 bone looks like. This is a 2, and this is a 3. So this is a way that you can transform qualitative information into something you can quantify. Yep, go ahead, Marcus.
Professor, can I ask you a question?
Yep.
So like when we do our manuscript and we're talking about like the morphology, would we want to show pictures and associate ratings with them? Or could it be like in our figure, or like in a figure or in a table caption, just be like rating 1 was like no bone loss, rating 2 was, how would you go about that?
Yeah, so you can spell it out in your methods section. And so you can spell it out in your methods section, and then it's up to you guys whether you would put representative images such as this into your methods, or you could combine these with like your graphs, right? So
okay,
just like this, we have our system or our scaling, and then this is the data we actually collected, basically. Okay. It would be up to you guys, but representative images are very helpful to your audience to be like, oh, this is how different, a 0 is versus a 3. Cool. Any other questions? All right. And so I'm going to skip this one too. We monitor GI transit. So like literally how much motility mouse GI tracts have following morphine exposure in our experimental compound. Same thing. If you guys have questions about this, feel free to ask. I'm happy to talk about it, but I think I've gotten the point across about using these things. So these slides are for you guys to use, or were for you guys to use when you're doing your morphology observations. So these are figure captions from this image. Again, it's basically just normal zebrafish anatomy so that you guys know what you're looking at when you're looking at them through the microscopes. So again, you guys have all these slide decks, so I'm not going to sit here and like read it off, but abbreviations, so FB, forebrain, MB, midbrain, forebrain, midbrain, hindbrain, etc. These were images for you guys to be able to identify zebrafish morphology when you were looking at it. Any questions about that? Okay. Again, another morphology slide, and just pointing out that 5 days post-fertilization and 7 days post-fertilization, larvae are very close in developmental structures. After 7 to 9 days post-fertilization, larvae enter the fry stage, where the yolk, which is right around here, yolk for Y, is depleted, and they begin to feed, right? So after 7 to 9 days, they actually need to start consuming food. And I think we feed them krill or some kind of shrimp. I could check with Aaron if anyone wants to know. I could check with Aaron if anyone wants to know, but essentially we are euthanizing our fish long before they get fed. So what they would get fed is not something you need to include in your husbandry data, again, because we're not actually feeding our larvae. You guys we're euthanizing them before they need food. Okay. And so again, more morphological notes for you guys to use in your slides. This is to help if anyone chose to use pigmentation as one of their morphological observations. This is just an example of that. Normal or typical pigmentation in a 5-day post-fertilization larva. And then so again, this is all kind of a moot point because we're beyond it. So these are representative images from a publication, right, to show you what normal versus dysmorphology would look like. And so up here, there's a zoom thing in my way. So this describes their treatment. This describes their treatment. And then they're basically ranking their scoring system down here, right? Part of this I can't see because of the zoom thing. Where am I? Still on tip chambers. Yeah, I mean, like I said, I'm not going to sit here and read this off. It was here for you guys to use. So these are representative images of jaw structure. These are representative images of like no inflated swim bladder versus a normal swim bladder, eye structure, jaw malformation between the two of these guys, heart, enlarged heart, I guess you want to call it. I know some people tried to look at the heart. And then excess yolk ball remnant. So just meant to show you guys that. Some people did use spinal, what do they call it? It's not spinal curvature. I mean, I guess you can call it spinal curvature if you want. So again, same thing. We have representative images, right, with how they would score on this scoring system. So this guy's normal. So on their normal scoring system, 5 is normal. You can technically, if you wanted to, you could have normal be 1. It doesn't really matter. So body shape score, this guy 5, somite morphology score 5. Body shape score 3, right? So we have slight dysmorphology here. Body shape 2, more severe than 3, obviously. And then incredibly severe, if you want to call it that. Body shape score 1, somite morphology score 1. Any questions about this stuff so far? Okay. We didn't use any transgenic reporter mice this semester. Sorry, not mice, reporter zebrafish. So we have several transgenic reporter zebrafish strains downstairs. We just don't have the equipment to essentially we don't have the equipment to allow you guys to use these zebrafish strains in mass. Like we don't have a scope that would allow 5 different groups to make observations like this all at once. And I believe I disseminated this information early in the semester. So, you know, if a group was really, really ambitious, we could have organized this. No one contacted me, so we haven't. So, you know, I think we have this strain of fish downstairs that alpha cells in the pancreas express green fluorescent protein. So we can image that. We also have the beta cell line. We also have the neuron line, right? Is this, so this is pancreatic development. This is we have this strain downstairs as well. So dopaminergic neurons express EGFP. Again, we have this line downstairs. We could have used it. I don't know if we can in 310. I don't know if we have the budget to like do more work. If this is really jiving with anyone, you know, shoot me an email and maybe we can make something work. But these are just representative images of, again, pancreatic labeling, dopaminergic neuron labeling. And then some extra resources for you guys. So the Zebrafish Information Network, link Zebrafish International Resource Center, link Zebrafish Anatomy Portal, link, and then Penn State Zebrafish Atlas. So again, if anyone wanted to look up more information about zebrafish anatomy or you know, find other information on normal versus not normal and scaling systems.